DNA and RNA differ only in a small way in terms of their structure. It's just the presence or absence of that 2' hydroxyl group. But that small difference in structure turns out to have a huge effect on reactivity. In particular, the 2' hydroxyl group in RNA opens up a reaction pathway for hydrolysis of the phosphodiester bond that isn't available in DNA. And in this webcast, we want to talk about those differences, and really what we're talking about is the enzymatic hydrolysis of the phosphodiester group. These are enzymes that are known as nuclease enzymes or phosphodiesterase enzymes. A typical example is actually found in the toxins in snake venom, in which we have an exonuclease, meaning that it cleaves nucleic acids from one end and travels along from one end to the other. In the case of the snake venom toxin, it's a 3' to 5' direction in which it travels. It actually digests DNA, not RNA, so obviously has toxicity to cells. Some nucleases have specificity, and these are restriction endonucleases. That means they cleave in the center of a nucleic acid with some kind of information specificity, such as maybe, as in the case of ribonuclease A, which we'll talk about in detail, following a particular kind of nucleobase. Ribonuclease A, for example, cleaves after the pyrimidines, the C or U. Ribonuclease A is an RNA cleaving nuclease. It cleaves and takes advantage of this reaction pathway that's made available by the 2' hydroxy group. So what it does, following a pyrimidine, is cleave the phosphodiester group, leaving a phosphate group as well as the hydroxyl group on the 5' position of the other side of the nucleic acid. It involves then the addition of water across that phosphodiester bond. And let's take a look at the mechanism of how this ribonuclease A takes advantage of the 2' hydroxyl group to open up a reaction pathway not available to the hydrolysis of DNA. It's a bifunctional catalysis mechanism, and we've seen many examples of these. Basically what that means in this case is there's a pKa modulation. The two histidines play off of one another so that one is going to be a base and one is going to be an acid. One's in its protonated form and one is in its neutral form. Also in the enzyme active site, there's a lysine, and we'll see that that helps to stabilize that trigonal bipyramidal intermediate in the phosphodiester hydrolysis. The way the mechanism gets started, right away we'll see how the 2' hydroxyl group comes into play. The most basic position, remember that's this sp2 position on the imidazole ring of histidine. That sp2 hybridized, or the N2 functional group, as we like to refer to that nitrogen as the N2 nitrogen, is the most basic site. It's going to deprotonate that hydroxyl group in a general base catalyzed way, which will allow it to act as an intramolecular nucleophile to the phosphodiester phosphorus atom. And that's what takes place in the first step. That's the addition step of the addition elimination pathway. Next, what we need to do is cleave this phosphorus oxygen bond to generate the 5' hydroxyl group on the exiting side of the nucleic acid that's cleaved in this reaction. How does that work? Well, we're going to use the general acid that's available from the imidazolium cation on histidine 119, and that's going to be the elimination process, a beta elimination process that eliminates this phosphorus oxygen bond, picking up the proton from that histidine on the imidazole ring of histidine 119 to make the hydroxyl group. You can see then that that leaves us with this cyclic phosphodiester, and in the next step that's going to undergo cleavage by addition with water. This negatively charged, this additional negative charge in the trigonal bipyramidal intermediate is stabilized by that lysine 41. That's the other key thing that I wanted to bring to your attention. All right, let's see how this cyclic phosphate undergoes hydrolysis with water. It's actually fairly strained, quite reactive compared to the normal phosphodiester group. The way that this happens is the presence of water in the enzyme active site is activated by a general base. Again, we'll use that most basic N2 type nitrogen to activate that as a nucleophile. It'll do an addition step. Again, that additional negative charge on this trigonal bipyramidal intermediate is stabilized by lysine 41. This then turns around and does a cleavage to leave the phosphomonoester on the 3' hydroxyl position, picking up a proton using histidine 12 as the general acid. That leaves histidine 12 in its original form, this neutral form, and histidine 119 has been left in its protonated form. We have the phosphomonoester on the end of the pyrimidine-containing nucleotide, and the result is we've cleaved the nucleic acid into two. So to summarize, basically the key difference, the key pathway that's opened up by that 2' hydroxyl group is this intramolecular nucleophile catalyst pathway that makes this cyclic phosphodiester intermediate. That pathway is not available to the DNA molecule, and so it's for that reason that RNA is about 1 billion or 3 billion fold more reactive than DNA. And the consequence of that is that DNA is essentially hardwired. That phosphodiester bond, as I mentioned, is extremely resistant. You can think of DNA as a hard drive, basically permanent storage of information, whereas RNA is only temporary carrying information. It's constantly turned over, and so the cell has capitalized on these very differences in reactivity to make very different functional use of these two information-carrying molecules.